Hello students, in today's lecture we will learn about photosynthesis in C3 and C4 plants. Let us now see the introduction. Photosynthesis is the only process in which many bacteria like cyanobacteria and eukaryotes like all algae and green plants can convert solar energy to chemical energy. During this process, solar energy is trapped and uses it to drive the synthesis of carbohydrate from carbon dioxide and water. There are three distinct events in the photosynthetic pathway. The first one is the light capturing events. Chlorophyll and the other photosynthetic pigments absorb specific wavelengths of light and excite the electrons present in the pigments. The second part is the light-dependent reactions. During these reactions, excited electrons from the light-capturing events are used to produce ATP with the release of hydrogen and oxygen. The released hydrogen ions will reduce NADP plus to NADPH. This reaction takes place in the grana of the chloroplast, but the NADPH and ATP leave the grana and enter the stroma for use of carbon fixation. The third is the carbon fixation cycle. These chemical reactions take the products, that is the ATP and NADPH of light-dependent reactions, and perform further chemical processes by fixing carbon dioxide from atmosphere. These reactions occur in stroma, which is the fluid-filled area of the chloroplast outside the thylakoid membranes. During these reactions, ATP and NADPH from the light-dependent reactions are used to attach carbon dioxide to ribulose bisphosphate or RUBP, which is a 5-carbon molecule, to manufacture a new and larger organic molecule like glucose. This process occurs only when light is available. Plants do not carry out the Calvin cycle during night time. There are three phases to the carbon fixation cycle, which is also collectively called the Calvin cycle. The first is the carboxylation of the carbon dioxide by the acceptor ribulose 1,5-bisphosphate, forming two molecules of 3-phosphoglycerate, the first stable intermediate of the Calvin cycle. The second part is the reduction of 3-phosphoglycerate, forming glyceraldehyde 3-phosphate, a carbohydrate. The next is the regeneration of the carbon dioxide acceptor, which is ribulose 1,5-bisphosphate from glyceraldehyde 3-phosphate. Let us now take up C3 photosynthesis. C3 carbon fixation is one of the three metabolic pathways for carbon fixation in photosynthesis, along with C4 and Chem. The plants exhibiting C3 photosynthesis or Calvin cycle are called C3 plants and includes about 95% of the total plants on Earth. Plants that survive solely on C3 fixation or C3 plants tend to thrive in areas where sunlight intensity is moderate, temperatures are moderate, and carbon dioxide concentrations are around 200 parts per million or higher. In C3 fixation, ribulose 1,5-bisphosphate or RUBP is carboxylated to form a 3-carbon compound via the activity of rubisco. During carbon fixation, carbon dioxide molecule condenses with ribulose 1,5-bisphosphate to produce a transient 6-carbon intermediate that rapidly hydrolyzes to two molecules of 3-phosphoglycerate or PZA. Because the first stable compound formed in this process is PZA, which contains 3 carbon atoms, this process is also called C3 fixation. The rubisco reaction forms part of a Calvin cycle that leads to the regeneration of ribulose 1,5-bisphosphate with the net production of glyceraldehyde 3-phosphate for the synthesis of sucrose and starch. 3 molecules of carbon dioxide must be fixed by the Rubisco to produce 6 molecules of PGA, which will generate 1 molecule of glyceraldehyde 3-phosphate, which is a triose phosphate. Following the formation of 6 molecules of 3-phosphoglycerate, 6 ATPs and 6 NADPH are used to generate 6 molecules of glyceraldehyde 3-phosphate. Only one of these molecules of glyceraldehyde 3-phosphate is then used to form fructose 6-phosphate and eventually starch. The other 5 molecules of glyceraldehyde 3-phosphate are converted in a series of steps involving 7 enzymes to 3 molecules of ribulose 5-phosphate, which are then phosphorylated using 3 ATP molecules to form 3 molecules of ribulose 1,5-bisphosphate, ready for another turn of the cycle. Hence, one complete turn of the cycle trapping 3 molecules of carbon dioxide as 1 molecule of glyceraldehyde 3-phosphate requires 6 plus 3, that is equal to 9 ATPs and 6 NADPH. Thus, the overall reaction can be represented as 3 molecules of carbon dioxide plus 9 molecules of ATP plus 6 molecules of NADPH plus 5 molecules of water gives rise to glyceraldehyde 3-phosphate plus 9 ADP plus 8 inorganic phosphate plus 6 molecules of NADP plus and 2 protons. Next, let us learn about C4 photosynthesis. The plants exhibiting C4 pathway are called C4 plants and comprises of 3% of flowering plants, which includes maize, sorghum and sugarcane, etc. They have approximately 50% higher photosynthesis efficiency than those of C3 plants such as rice, wheat and potato. This C4 pathway is also known as Hatch and Slack cycle and is named C4 for the 4-carbon molecule, which is the oxaloacetate formed in the first product of carbon fixation in the plants that use that process. In contrast to the 3-carbon molecule PGA produced in C3 plants. The C4 photosynthetic carbon cycle is an elaborated addition to the C3 photosynthetic pathway. This C4 carbon fixation is evolved to adapt high light intensities, high temperatures and dryness. So, C4 plants dominate grassland floras and biomass production in the warmer climates of the tropical and subtropical regions. For C4 plants, photosynthetic activities are partitioned between mesophyll and bundle seed cells that are anatomically and biochemically distinct. Under normal atmospheric conditions, Rubisco adds carbon dioxide to RUPP. However, when the carbon dioxide concentration is low, it can add oxygen instead. This produces phosphoglycolate and 3-phosphoglycerate, and the process is known as photorespiration. Phosphoglycolate has no known metabolic purpose, and in higher concentrations it is toxic for the plant. Photorespiration is not only energy demanding but furthermore leads to a net loss of carbon dioxide. Thus, the efficiency of photosynthesis can be decreased by 40% under unfavorable conditions including high temperatures and dryness. Plants develop different ways to cope with this problem of photorespiration. C4 photosynthesis is one of the most successful ways evolved by plants to reduce photorespiration. C4 and CHEM pathways minimize photorespiration, which wastefully fixes oxygen rather than carbon dioxide. C4 plants conduct C4 photosynthesis in mesophyll cells and Calvin cycle in bundle sheath cells. Rubisco located in bundle sheath is isolated from atmospheric oxygen, which is found in mesophyll cells. The carbon dioxide for Calvin cycle is shuttled via malate from mesophyll to bundle sheath, where it is released again through decarboxylation of the malate. C4 plants use PEP carboxylase to capture more carbon dioxide in the mesophyll cells. Instead of using carbon dioxide in mesophyll cells, PEP carboxylase utilizes bicarbonate. The carbon dioxide entering the mesophyll cells from the atmosphere will be converted to bicarbonate by hydrating carbon dioxide using carbonic anhydrase. This bicarbonate will bind with phosphoenol pyruvate or PEP in presence of PEP carboxylase to form oxaloacetate. Oxaloacetate is the first stable compound having 4 carbons, so the pathway is called C4 pathway. Then, oxaloacetate is reduced to malate or aspartate, which are 4 carbon compounds. The malate or aspartate enters bundle seed cells and releases carbon dioxide inside bundle seed through decarboxylation. The released carbon dioxide is immediately fixed by Rubisco, which will be followed by normal Kelvin cycle. The remaining 3 carbon compound pyruvate formed after decarboxylation of malate will return to mesophyll cell for next C4 cycle using energy in the form of ATP. Photorespiration is a major problem for plants in hot climates as it consumes oxygen and releases carbon dioxide. During hot conditions, the plants close the stomata to conserve water, but this leads to a drop in the carbon dioxide concentration within the leaf, favoring photorespiration. In addition, as temperature rises, the oxygenous activity of Rubisco using oxygen increases more rapidly than the carboxylase activity using carbon dioxide, again favoring photorespiration. To avoid these problems, some plants adapt to live in hot climates such as corn and sugarcane have evolved a mechanism to maximize carboxylase activity of Rubisco. In C4 plants, the carbon fixation activity of PEP carboxylase is physically separated from the carbon reduction process by Rubisco. Rubisco is produced by mesophyll cells in C3 plants, but mesophyll cells of C4 plants produce PEP carboxylase, where assimilation and fixation of carbon occurs. Carbon reduction by Rubisco, which is isolated from mesophyll cells, occur in the cells surrounding the vascular bundle known as bundle seed cells. Since the bundle seed cells are not directly exposed to air, the oxygen concentration is low. The carbon dioxide is transported from the air via the mesophyll cells to the bundle seed cells by combining with 3 carbon molecule pyruvate to produce 4 carbon molecule oxaloacetate, which will be reduced to malate. These enter the bundle seed cells where they are broken down to C3 compounds, releasing carbon dioxide. At the end, the initial substrate PEP of the C4 cycle is regenerated in mesophyll cells through pyruvate. Thus, the C3 molecules return to the mesophyll cell to accept more carbon dioxide. This cycle ensures a high carbon dioxide concentration for the carboxylase activity of Rubisco action in the bundle seed cells. Let us now see the comparison of C3 and C4 cycle. C4 improves photosynthetic performance by forcing carbon dioxide into the standard C3 photosynthetic apparatus. The carbon dioxide concentration mechanism of C4 plants suppresses the oxygenation reaction by Rubisco and the subsequent energy wasteful photorespiratory pathway. C4 plants are more successful than C3 plants in areas with high light, low water and low nutrients as they make more efficient use of available light, particularly at high temperatures. In full sunlight, C3 plants use only a fraction of the available light energy to convert to carbohydrates, while C4 plants continue to turn carbon dioxide into carbohydrate as available light increases. C4 plants also make better use of water and nitrogen than C3 plants do. In C4 plants, transpiration is also reduced as they can maintain carbon dioxide concentration even when keeping their stomata closed. Due to production of more biomass and higher photosynthetic rate per unit of nitrogen, C4 plants can grow and reproduce even on nitrogen-poor soils as compared to C3 plants. Let us now see the conclusion. Most of the plants show C3 mechanism for carbon fixation during photosynthesis. However, one of the most important enzymes involved in carbon fixation, that is rubisco, has both carboxylase and oxygenase activity. And during certain conditions, instead of acting as carboxylase, rubisco acts as oxygenase, leading to photorespiration that results in loss of huge energy. To avoid additional loss of energy through photorespiration, plants have evolved to C4 mechanism, which avoid the losses during photosynthesis. Although C3 plants are more common in nature than C4 plants are found to have more photosynthetic activity due to more efficient use of carbon dioxide and light. Understanding the mechanism of C3 and C4 will help in the increasing of productivity in plants. Thank you.